Assalamualaikum warahmatullahi wabarakatuh to Shadin. Today we're going to talk about genetic engineering and our topic is introduction and tools used. Our group members is Hasril, Yusuf, Alia, Farisha, Humaira and me, Alia. So genetic engineering is the process of using recombinant DNA technology to alter the genetic makeup of an organism. Okay, next slide please. Okay, so as you can see, this is our poster. So our topic is introduction, tools, and tools used in genetic engineering. So there's two components, which is the introduction and discussion that we're going to talk about. In the discussion, there's two tools that is being used, which is enzymes and vector. So there's 4 types of enzymes and 4 types of vector that is going to be elaborated by my fellow groupmates. So there's steps of gene cloning. Okay, the first step is isolation of DNA of interest. Next is ligation. Third is transfection, or also known as transformation. And fourth is screening and selection procedure. Okay, so we're gonna take a look on the diagram of this. So next slide, Hasril. As you can see, this is the step of cloning. Okay, first is in order to clone a fragment of DNA, we need to insert it into a vector that will replicate in a host cell. Okay, first step is the isolation of DNA of interest. Okay, as you can see this, in this poster, this is a foreign DNA and this is a plasmid. Okay, so in the plasmid, there's three different components, which is the ampicillin resistance gene, the LAC-Z gene, the restriction site, and then there's the foreign DNA. Then we have to use a restriction enzyme to cleave a section of the desired DNA fragment in the foreign DNA by using a restriction enzyme. So, and then we have to use the same restriction enzyme to cleave at the plasmid too. So why do we use the same restriction enzyme? We use the same restriction enzyme because they cut a specific sequence. So the same restriction enzyme must be used so that we have the same complementary sticky ends. So by cutting the DNA and the plasmid, it produce sticky ends. As you can see here, sticky ends. So these sticky ends will combine together and form a recombinant plasmid. So that is in the next step. It's called ligation. So in this step, since now both the DNA fragment and the plasmid have sticky ends that complement each other, the DNA fragment can be inserted into the plasmid. So it will turn into recombinant plasmid. So this recombinant plasmid will be mixed with DNA ligase, and the DNA ligase will reattach the DNA backbones. So so that it produces recombinant plasmid. Okay, the third step of cloning is called transformation. So in this step, the recombinant plasmid is inserted into a host cell. There. So normally the host cell would be a bacteria or some other organism that can allow it to grow. So first, we have to know that not all the bacteria would take up the recombinant plasmid. Most of them would not take up the plasmid. Most of them also will not take up the plasmid, but the plasmid doesn't have the foreign DNA. That means it's just a normal plasmid like this one. So then only a few will take out the recombinant plasmid inside the bacteria. So once they take out the recombinant plasmid, then we can grow them up. And this is in the next step. So when we grow them, we would not know which bacteria contains the plasmid and which bacteria does not contain plasmid and which bacteria contains plasmid but not the foreign DNA. So when we grow them, we have to, in order for us to have the bacteria that has the plasmid that we want, like this one, we have to go through selection process. So this is the first step, selection process or screening process. So in the selection or screening process, the solution containing both the bacteria and also plasmid is poured into an agar that has nutrients and also ampicillin, which is the what do you call that, antibiotic. So when we put the bacteria inside the agar that has nutrients and ampicillin, the bacteria that has plasmid will survive, and the bacteria that does not have plasmid will die. This is because in the plasmid we have the ampicillin resistance gene, so it can resist the antibiotic. So that means it can grow. It can grow. It can be cultivated. And then next, once we find out that once the bacteria that does not contain the plasmid die, we also have the bacteria that contains the plasmid and the bacteria that doesn't contain that the bacteria that contains plasmid but doesn't have the foreign DNA. So then we insert this into the agar medium. The agar medium provides two different colors, which is blue, blue and white. So the white ones is the colonies that have plasmid with the foreign DNA, and then the blue one is the one that doesn't have plasmid. So once we differentiate the blue one and the white one, we take out the white one, and then we have to go through the next stage, which is colony hybridization. Once we take out the white one, we can cultivate the colony hybridization. Colony hybridization is when we grow all the white colonies. So what we have is just we're left with the bacteria that have the plasmid and the foreign DNA. Okay, that's the steps of DNA cloning. Next, please, Hasril. So the last step is the methods of cloning. Methods of cloning, there's two ways, the in vivo and in vitro. In vivo is the one that I talked about just now, which is they use the restriction enzyme and ligase and using vectors and cloning fragments into host cells. While the PCR in vitro is when they use polymerase chain reaction, which is PCR. They only create copies of fragments of DNA. That's all from me. I would like to pass this presentation to Hasril for the next topic.
Thank you, Alia. Moving on, genetic engineering relies on enzymes, two types in particular, which I'm going to explain. The restriction enzyme and also DNA ligase. So let's take a few minutes to see the roles of these two enzymes in genetic engineering. We can say that one of these enzymes kind of acts like scissors. It is known as a restriction enzyme, and its job is to cut DNA at particular sequence. And then there's another enzyme known as the ligase, DNA ligase, and its job is to glue DNA back together. This is DNA ligase, and this is the restriction enzyme. Now let us explore restriction enzymes in a little bit more detail. There are many different restriction enzymes, thousands of them. Each enzyme is specific for a particular DNA sequence. So this restriction enzyme will only cut DNA when it sees the sequence, for instance, in this diagram, CCTGG. And this enzyme will only cut DNA when it sees the sequence TTCGAA. And this enzyme will only cut DNA when it sees the sequence CCTAGG. So and so forth. The other thing to know about restriction enzyme is that many of them cut unevenly, and this is known as the restriction site. So let's take a closer look on how the restriction enzyme cuts unevenly. So here is a DNA sequence, and here is the restriction site for the particular enzyme. It likes to start by cutting here, but then finish cutting here. So what happens is that we get an uneven DNA sequence. These uneven DNA sequence are known as sticky ends. And they are sticky ends because since there is only one DNA sequence exposed here, it can easily bind through complementary base pairing rules to another sticky end. And this is important to genetic engineering. So here we've got our sticky ends. Now let's take a closer look at how we would use these two enzymes mentioned earlier, which are the restriction enzymes and DNA ligase together in order to create a recombinant DNA. So here's our DNA sequence and here's the restriction site for a particular restriction enzyme. First thing that we're going to do is add a restriction enzyme to cut it into fragments, hence producing the sticky ends. And now we're going to add a DNA from another source that has also been cut using the same enzyme. This is important because by using the same restriction enzyme means that you get the same sticky ends. So here is DNA from one organism and here's DNA from a different organism, but the same sticky ends. Now these fragments are going to stick together because of complementary base pairing. So here we go. We got this sticky end is complementary to the sticky end and the sticky end is complementary to the sticky end. So they naturally came together. However, this is where ligase is needed at this point because ligase will help attach the sugar phosphate backbones, which are more difficult to make. So once ligase gets involved, and here we go, we now have our recombinant DNA. DNA from one organism combined with DNA from another organism. Therefore, this is how we would put two different genes together. But at this point, it is pretty useless because all we have is a recombinant DNA molecule. And in order for that to actually build a protein that makes a trait, we need to put this DNA into an organism, which will be discussed by my other group mates. And that is all from me. I will pass to Alia to continue with her part.
Okay, so thank you so much to Hasril. So next, I'll be talking about the third and the fourth tools that is used in the genetic engineering. And the first one is the DNA polymerase. Okay, DNA polymerase is a group of enzymes that synthesize complementary nucleic acid molecules to the template. What I mean by a group is there's actually 3 DNA polymerase that is used in DNA replication that we learned in the previous chapter, which DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. So the function of DNA polymerase 1 is actually they have exonuclease activity that removes RNA primer and they replace with the new DNA. And DNA polymerase 2, their function is just to repair any misreading on the DNA. And the last one is the DNA polymerase 3. It's the main enzyme that adds nucleotide in 5 to 3 direction to the template. Okay. However, there are 3 types of polymerase that are used in genetic engineering. The first one is the DNA-dependent DNA polymerase, which it replicates DNA from another DNA. And then the second one is the DNA-dependent RNA polymerase. They transcribe RNA from a DNA. And the last one is the RNA-dependent DNA polymerase, which is it's actually the reverse transcriptase because it reverse transcribe DNA from an RNA. Okay, next slide. Okay, so the first one is the reverse transcriptase. Okay, reverse transcriptase, also known as the RNA directed DNA polymerase, as I said earlier. This enzyme is usually they encoded from the genetic material of a retrovirus because it can generate complementary DNA, which is cDNA, from an RNA. And they also can be found it can cause disease such as HIV and AIDS. It also be used as tools to make an antiviral drugs, which the antiviral drugs can inhibit the reverse transcriptase. Okay, also in the lab, this enzyme also called as the reverse transcription polymerase chain, which is RT-PCR. It can be used to diagnose disease such as cancer, and also uses for to do a research. Okay, that's all from me. Next, I'll let Farisha to continue the presentation.
Thank you, Alia. So I'll be explaining about the vectors. The second step in molecular cloning is to join the passenger DNA to a DNA of stable cloning vehicle, or we call vectors. Vectors are DNA molecules that carry DNA fragments from one organism to another. It allows the insertion of exogenous DNA into a bacterial target. The vectors are commonly used are bacterial plasmids and bacteriophage DNA. So I'll be explaining about the plasmids and bacteriophage. Next slide, please. After restriction digestion, genes of interest are commonly inserted into plasmids. In bacterial cells, plasmids are small pieces of typically circular double-stranded DNA that replicate independently of the bacterial chromosome. These plasmids are distinct units from the larger portions of DNA that make up the bacterial genome. These plasmids can easily be separated on the basis of size. So, plasmid vectors used for cloning typically have a polylinker site or multiple cloning site. A polylinker site is a short sequence containing multiple unit restriction enzymes, mention sites that are used for inserting DNA into the plasmid after restriction digestion of both the DNA and plasmid. Having these multiple restriction enzyme recognition sites within the polylinker site makes the plasmid vector versatile, so it can be used for many different cloning experiments involving different restriction enzymes. This polylinker site is often found within a reporter gene, another gene sequence artificially engineered into the plasmid that encodes a protein that allows for visualization of DNA insertion. So first, the bacterial cell are first homogenized and chromosomal DNA is centrifuged down, leaving the plasmid DNA in the supernatant. The plasmids are then isolated and purified before cutting with a restriction enzyme. If a restriction enzyme has been used to isolate the required gene, the same restriction enzyme must be used to cut the plasmid DNA. The restriction fragments from the donor DNA containing the wanted gene are mixed with the plasmid DNA and joined either by their sticky ends or blunt ends, depending on the type of restriction enzymes used. So there are several properties of plasmid. First, it is small. Why it must be small? Because it contains minimal DNA material and it is easy to purify because they are less fragile. Second, it has no DNA sequence, so it is easy for genetic engineers to manipulate. Third, it contains a selectable gene marker. Next, it contains a second selectable gene marker, genes that code for beta galactoside, which can break down the colorless X-gal to a blue compound and inactivated by insertion of a passenger DNA. Next, it has many unique restriction sites, so it allows flexibility in cloning. It also has rapid replication in host cell, so it gives higher yield but less energy. Next, it is origin of replication. It allows plasmid to replicate within and semi-independently of the host cell. Next slide. So the arrows in plasmid, the arrows indicate the directions in which the genes are transcribed. Note that polylinker site containing multiple unique restriction enzyme recognition sites found within the LEC-Z reporter gene. Also note that the ampicillin resistance gene encoded on the plasmid. Next slide. Next slide. Okay, so my next part is bacteriophage vector. Bacteriophage are viruses that replicate within the bacteria. Phage vectors can accept short fragments of foreign DNA into their genome. They can undergo two life cycles, which are lytic cycle and lysogenic cycle. Examples are lambda phage, M13 phage, T4, T7 fish, and etc. So next slide. So one of the example of bacteriophage is fish lambda. It has head, tail, and tail fibers. Its genome consists of 48.5 kb of DNA, which comprise of sticky ends at both the terminals. Since these ends are complementary, they are cohesive and also referred to as cosites. So why bacteriophage as a vector? It can accept very large pieces of foreign DNA. Fish that have a stuffer fragment are called substitution vectors because they are designed to have a piece removed and substitute with something else. But why clone large pieces of DNA in the first place? So one obvious reason is that some genes are very big and it's advantageous to have them in all one piece. Another reason for cloning in lambda is the efficiency it offers in DNA transformation. So that's all from me. Thank you.
So I am going to talk about the third vector, which is YAC. So YAC is actually a plasmid shuttle vector that is classified as an artificial chromosomes in tools for genetic engineering. So shuttle vector means it is capable of replicating in both yeast and also bacteria. So YAC and BAC is a human engineered DNA molecule that derive from DNA of the yeast and also common bacterial hosts such as E. coli, which is then ligated into bacterial plasmid or yeast plasmid. So they act as a cloning vehicle that will breed in eukaryotic cells hosted as eukaryotic chromosomes. Okay, I'll move on to the function. The first function is it is a cloning vehicle. Second function, it provides the largest insert capacity of any cloning system. This is because a YAC it is used to clone DNA fragments that is larger than 100 kb and up to 3000 kb. And then the third function is physical mapping and genomes. So segments of an organism's DNA, which is up to 1 million base pairs in length, can be inserted into the YACs. Then the YACs with the inserted DNA will then be taken up by yeast cells. So as the yeast cells grow and divide, they amplify the YAC DNA, which can be used and isolated for DNA mapping and sequencing. I'll move on to the advantage and the disadvantages. So the first advantage is that it is very easy to find and cheap. So genetic manipulation in yeast is much more easy and cheap compared to similar experiments in a more complex animals such as mice or zebrafish. And then the second advantage is that it is stable in prokaryotic systems. So when it is cloned into prokaryotic system, it remains stable and intact in YAC clones. Although it may seem that yeast and humans have little in common, yeast is actually a eukaryotic organism. So it means that just like our cells, it has a nucleus that contains DNA packaged in the chromosomes. So the third advantage is that yeast cells share many basic biological properties with our cells. So the gene can be used to test new drugs on human genes to see if the drug can restore the normal function. And then we'll move on to the disadvantages. The first disadvantage is that it is less stable as YAC clone a lot of DNA fragments. So YAC clones frequently contain deletions, rearrangements, or non-contiguous pieces of the cloned DNA, and as a result, each YAC clone have to be carefully analyzed to make sure that no arrangements of DNA occur. And then the second disadvantage is that the efficiency of cloning is very low, which is just about 1,000 clones that are obtained per microgram of vector and insert DNA. Next slide, please. Okay, I am going to talk about the differences between YAC and BAC. So the first one is that the recombinant vectors of YAC will transform into yeast cells. Meanwhile, in BAC, it will transform into bacteria. And then in YAC, it will produce based on specific regions of the yeast chromosomes. And in BAC, it is produced based on the F plasmid. And then the DNA of YAC is linear, while BAC is circular. And then the four differences is that a single vector occur in YAC, there is only a single vector that occur per yeast cells, while in BAC, there are 1 to 2 vectors that occur per bacterial cells. And then the last differences is that YAC is high cloning capacity, which can be insert up to 1000 kb, and BAC is has lesser cloning capacity, which it can only insert up to 200 kb. Next slide. Okay, next I'm going to talk the last vectors, which is cosmid. So cosmid have been constructed to include a fragment of lambda that includes the cos sites. So cloning into cosmids is very, very similar to cloning in lambda phage, and it involves digesting exogenous DNA with a restriction enzyme, cutting them, combining them, and ligating them. And cosmid vectors are very useful tools for establishing gene libraries for a number of different prokaryotic and also eukaryotic species. So it is just like plasmids, but it just have multiple cloning sites, 2 cos sites to be exact. I am going to move on to the function. So the first function is that it is used to build genomic libraries. The second one is that it is used to clone large fragments of DNA because it can carry up to 45 kb. And the third one is that it can be used in homologous recombination between 2 different plasmids in the same cell and grown in both bacteria and also animal cells. And then the advantages and the disadvantages. The first advantage is that cosmid has quite large capacity of DNA fragments, which the cosmid vector can carry up to 45 kb, whereas plasmid and lambda phage vectors are limited to only 25 kb. The second advantage is that it has high transformation efficiency. The third advantage is that it have easy screening method. And then the disadvantage is that the genomic DNA fragments sometimes join together in the ligation reaction. This is because when it clones a large fragments of DNA, a lot of time during a partial digestion with restriction enzymes, 2 or more genomic DNA fragments will join together in the ligation reaction. So this will cause fragments that were not initially adjacent to each other. That's all for me.
Hi, Assalamualaikum. For my part, I'll be explaining on the components of the YAC and the cosmids. So I will be explaining on why the components are important and essential for the vectors. So as what was just stated just now from Myra, well, the YAC is a cloning vector used to clone DNA fragments from several mega base pairs up to maybe a lot of mega base pairs. So the features in the YAC, we can find the first one is the centromer sequence. The centromer sequence are essential for it to propagate inside the yeast, whether it is a bacteria or yeast. The centromer sequence is essential for propagation. So what Myra just said just now, YAC is like a shuttle vector. It can shuttle between yeast and bacteria. That's why it has bacterial replication as well. So the next one is telomere sequence. It is important for the stability of chromosome inside the eukaryote. And we can also find in the components, we have the selectable markers or the selectable amplification. At the end, it has a multiple cloning site, which is common features for any cloning vector. So strategically, there should be 2 BAMH1 sites placed flanking the telomeres, which is to digest and restrict a certain fragment, thus making it circular, thus making the circular vector linearized. The linear vector can then be restricted into other enzyme with another enzyme in the MCS, that is EcoR1. The fragmented site can then be added with gene DNA fragments of interest. So moving on to the next one, the cosmate. So when we look at the name itself, cosmate is actually cosites plus plasmate. Cosites are also Humaira just stated just now. So in a bacteriophage, a bacteriophage can infect bacterium efficiently. At the end of the phage genome are very sticky. We can find very sticky substance. Those sticky substance are the cohesive site that help package inside the heads of the phage and to make sure that they circularize properly inside the bacteria. If the cosites are 50 kilo base pairs away, then the efficiency for packaging inside the phage head is very efficient. Otherwise, no packaging takes place. So the features for the cosmates are modified versions of plasmids. It has multiple cloning sites like any other cloning vector. Cosmates can carry 37 to 52 and normally 45 kilobase pairs of DNA. And the origins of replication so that it can be propagated inside the bacteria. And the cohesive sites on the plasmids allows circulation inside the bacteria, as well as the restriction genes, which allows selection of the recombinants inside the bacteria. So moving on to the conclusion. In conclusion, genetic engineering is a scientific advancement in mankind that has brought biotechnology innovations. Genetic engineering can pose a lot of hazards, but once it is understood that everything has the potential to be dangerous or damaging, the fears towards genetic engineering will be reduced. So that's all from us. Thank you for listening.